We did the grain size distribution lab. Next slide. The grain size analysis test is used to approximate the individual grain size distributions of a soil sample. For this, particles are categorized into a range of sizes. There are two different tests we use to determine the grain sizes. The sieve analysis is used to measure the sizes of larger particles, while the hydrometer test is used to measure the finer particles. The objective of this lab was to create a size distribution chart with data from both tests, and then determine what type of soil it is based off the USCS soil classification chart.
Grain size distribution is a major component of soil classification using the Unified Soil Classification System, USCS. The standard grain size analysis test determines the relative proportion of different grain sizes as they are distributed among the certain size ranges. Larger particles testing is accomplished using sieves. Sieve analysis allows you to directly measure the mass and retained and mass passing a specific diameter particle size. These values can be used to calculate percent retained and percent finer in order to construct a grain size distribution graph. The hydrometer test enables you to determine the clay fraction of the soil. Although not necessary for USCS classification, it is used to better develop the grain size distribution for the finer portion of the soil. The first step in the sieve analysis procedure is to snap together sieve pans in ascending size order, smallest pan on the bottom. The second step is to obtain a soil sample of at least 500 grams. From ASTM D422, we obtain a soil sample of at least 500 grams if the nominal diameter of largest particle in the soil being tested is 3/8 of an inch. We then place lids on the sieve stack and shake for 3 to 5 minutes. Then we remove the stacks from the shaker and weigh each individual sieve. Once the sieves have been weighed, we deposit the soil back into the bin and set the number 10 through number 200 sieve aside for further testing. The first step in the hydrometer testing procedure is to obtain at least a 60-gram sample of air-dried soil that has passed the number 10 sieve and place it in a 250 milliliter beaker. We then thoroughly mix 125 milliliters of sodium hexametaphosphate solution with the soil in the beaker. Next, we transfer the beaker contents into a dispersion cup, making sure to wash the entire sample into the cup. Disperse the sample for 1 minute using the mixer. Next, transfer the sample from the cup into a 1,000 milliliter graduated cylinder, making sure to wash the entire sample into the cylinder. Fill the cylinder to 1,000 milliliter mark with distilled water. Another 1,000 milliliter cylinder with control solution will be provided for each group. Record the temperature and the hydrometer reading in this solution. This hydrometer reading will be the composite correction reading. Cover the top of the cylinder and repeatedly invert it to further disperse the soil in the solution for 1 minute. Place cylinder on table and start the stopwatch. For each reading, carefully insert, but do not drop, the hydrometer into the solution and read the value once the hydrometer has stopped bobbing. Also record the temperature at each reading. Place the hydrometer in the control solution between readings. Afterward, perform the calculations and generate the grain size distribution graph.
For the sieve analysis, a total mass of 595.7 grams of a soil sample were used. The mass of the sieves were recorded before the lab began. The mass retained on each sieve was determined by subtracting the mass of the sieve from the combined mass of the sieve and the soil that was retained on it. The percent passing was determined by dividing the mass by the total amount of soil. In this case, we found that 95.9% of the soil passed the number 4 sieve, while 10.9% of the soil passed the number 200 sieve. For the hydrometer, a total of 64.8 grams of soil were used with a specific gravity of 2.75. This soil was what was left in the pan of the sieve analysis test. 8 measurements were recorded for the listed times. A reference reading of 1.001 was used to calculate the actual hydrometer reading. The variables L and K were taken from tables 9-3 and 9-4 respectively from ASTM D422-63. Next slide. Our final grain size distribution curve is shown here. Overall, the soil sample was comprised of 4.1% gravel, 85% sand, and 10.9% fine particles. Of the fine particles, none of them were smaller than 0.002 millimeters in diameter, and therefore did not qualify as clay. Based off the previous grain size distribution chart, 60% of the soil passed 1 millimeter, 30% passed 0.35 millimeters, and 10% passed 0.075 millimeters. Using those in calculations, we determined that the soil was a well-graded sand.
The sieve analysis and hydrometer tests were used in conjunction with one another to determine the makeup of a soil sample and where it lies on the Unified Soil Classification System, or the USCS chart. The sieve analysis works by shaking a soil sample and allowing grains to either pass or be retained by a number of graduated pan sizes. The hydrometer test is a timed measure of how long it takes soil grains to settle when suspended in deionized water. To firstly address the sample that was tested, a sieve analysis was conducted to determine the makeup of the sample and what percentage of the sample surpassed the number 200 level sieve. Looking at the sieve number 4, 96% of the sample passed, marking the sample as a sand. Looking at the sieve number 200, approximately 11% of the sample passed. Based upon this passing percentage, we can conclude that the sample is a coarse-grained soil. Upon further classification using the USCS table, 11% of the soil was made up of fines. This qualifies it as a sand with fines. From here, we must use the hydrometer test to answer further questions regarding sand classification. The hydrometer test will explain how to proceed now that we have reached a threshold that lies between the given parameters of 5 and 12% fines stipulated in the USCS. We will now have to evaluate the coefficient of uniformity and the coefficient of curvature to determine whether or not there is a W or P and G or S appended as a dual symbol. Based on the hydrometer test, the necessary symbols for SW or SWSM, making the sand either well-graded or silty. From here, we can refine even further using the calculation for the coefficient of curvature, which was determined to be 2.833, and the coefficient of uniformity, which was determined to be 11.333. It is most likely that the soil sample is a well-graded sand. On the next slide, we see that the Texas Department of Transportation conducted a similar experiment in 2016, in which they tested a soil sample under a sieve analysis and hydrometer test. The experiment differed slightly, firstly in their sieve analysis, in which only 4 pans were utilized, number 4, 10, 40, and 200. 91.9% of their sample was retained by the number 200 sieve. This indicates that they were then placed into a similar disadvantage as their fines lay between 5 and 12%. The issue with this is that their broadness and scale has sacrificed specificity, as they are unable to calculate a reliable coefficient of curvature or coefficient of uniformity. Based upon their measurements, the sample can then be categorized as a poorly graded gravel. The immediate difference between these samples is evident, as the NCSU's tested sample was a sand and the Texas Department of Transportation was a gravel. On the next slide, we can see the USCS ASTM flow chart, where we can categorize firstly the gravel that was tested by the Department of Transportation in Texas, as we can categorize firstly by the fines, then by the coefficients we broadly determined based upon their pan sizes to a poorly graded gravel with silt, and then ours as we move down the flow chart to define it as a well-graded sand with silt. On the next slide, based upon the data granted from both labs, it is generally understood that the sieve analysis and complementary hydrometer tests provide adequate measures by which to analyze and categorize a soil sample. The two work in tandem to decently estimate the average grain size diameters on a scaled distribution. The sieve analysis determines the component breakdown, while the hydrometer test determines the grain size aspects through dispersion. From this data, coefficients of curvature and coefficients of uniformity may be determined. Based on these factors working in conjunction, the soil sample is able to be categorized. This test is far more reliable than the specific gravity test, as it grants a larger range of values to parse. The greater availability of data allows for a more accurate sample determination. Testing more samples of the same source material will help mitigate margins of error. From the data provided to us, we determined the sample to be a well-graded sand with silt. Due to its simplistic procedure and straightforward results, the grain size distribution tests are our choice of experimentation in order to determine the makeup of a soil sample.